In a neonatal intensive care unit at the Children's Hospital of Philadelphia, a toxic molecule, ammonia, is rising in newborn baby KJ's blood. For parents Kyle and Nicole Muldoon, a nightmare scenario. Doctors know exactly what's happening to their son, but not how to fix it. CPS1 deficiency is a rare genetic disease that keeps the body from properly breaking down protein, with catastrophic results. Half of those affected die in infancy. Survivors tend to suffer severe neurological damage. For KJ, the prognosis is grim. He's being kept alive by dialysis, ammonia-scavenging drugs, and 24-hour care. Parents Nicole and Kyle are preparing for the unthinkable, holding on to a thread of hope.
It took at least as much effort to do the blackjack playing mechanics and to remember all the numbers and everything as it did to be aware of all your surroundings and to try to stay ahead of casino security. We were communicating non-verbally by how we held our drinks. We would often have our phones set to call us 40 minutes after we would sit down to minimize the chance that the pit had the time to do a skill check on us.
In 2012, biology researcher David Liu was teaching a class at Harvard, an unofficial curriculum in probability, advanced math, discipline, a hands-on course on beating the casino.
We met every Friday in my office. I simulated billions and billions of hands of blackjack. That turned into a system that two people could actually play at an advantage over the house.
Over 4 years, Liu and his team of students would amass an undisclosed amount of winnings in Las Vegas before the house finally caught on.
We get surrounded by suits and they say, "You're no longer welcome to play blackjack." I was backroomed twice. We were barred from almost all the major casinos in Las Vegas. We made a lot more money than we were statistically supposed to.
Ultimately, Liu's passion for untangling complexity would land him on a path with stakes higher than any casino game. After all, in his day job, Liu was a Harvard molecular biologist.
I really was awed by the power of DNA and the ability of the molecules of life to evolve, to replicate, to mutate, to undergo Darwinian selection is really what drives all of biological evolution.
In DNA, 4 nucleotide molecules, each represented by a letter, A, G, C, or T, make up the string of instructions over 3 billion letters long that contain all the information on how to build and operate our bodies. But random mistakes, or mutations, happen when DNA makes copies of itself. Mostly, the changes don't matter. Sometimes a mutation can lead to an evolutionary advantage, and sometimes even a single letter error in the right place can have devastating consequences.
Thousands of genetic diseases result from relatively small mutations in DNA. Sickle cell disease, progeria, cystic fibrosis, and a host of others afflict an estimated 400 million people worldwide, with few effective treatments at hand. While doctors understand the causes of such conditions, they typically can't correct them. Liu's research with organic molecules and DNA dated back to the mid-90s, but a discovery some 2 decades later had reinvigorated Liu's curiosity. The new technique called CRISPR gave researchers the unprecedented ability to precisely cut DNA.
The problem that I became increasingly interested in was how to use the beautiful search mechanism of CRISPR to engage a target DNA sequence.
Liu imagined a tool that could fix DNA mutations, a molecular pencil that could erase and rewrite the code. This tool would have to work not only in theory, but under real-world conditions.
The lab got to work, with Liu and students painstakingly dissecting the fragile systems of DNA synthesis and seeking out ways to exploit the system. In 2019, that knowledge crystallized into a world-changing discovery, one that would allow them to make precise, targeted changes to the genetic code, the world's first DNA base editor.
Base editing works like sort of molecular pencils that rewrite a single DNA letter to become a different DNA letter.
A pencil to correct typos in the code of life, the very same typos that cause rare diseases, like CPS1 deficiency. When KJ Muldoon's diagnosis was confirmed, 2 physician scientists, CHOP's Rebecca Ahrens-Nicholas and UPenn's Kiran Musunuru, moved quickly, proposing a radical idea: to use Liu's base editor in a desperate attempt to save KJ's life. While CHOP's intensive care kept KJ alive, they shaped Liu's discovery into a bespoke gene editing tool programmed to correct KJ's DNA. With the clock ticking, teams of scientists fast-tracked every step of a process that had never been attempted before. Even FDA approval, a process that normally takes years, came within 6 months. In February of 2025, KJ received the first of 3 injections. Within days, his symptoms started to improve, and for the first time, at 10 months old, KJ sat upright.
KJ was dosed less than 7 months after birth with a customized base editor, and the result is that baby KJ is thriving now.
In June, after 307 days at CHOP, KJ went home.
Having the opportunity to work with such an incredibly talented group of students and postdocs and collaborators, that's really what makes me pinch myself every morning that I get to do the job that I have.